Good afternoon and welcome to all. Today we are going to discuss about bacterial reproduction. Actually, these bacteria are the prokaryotic organisms that reproduce asexually. And the bacterial reproduction most commonly occurs by a kind of cell division called binary fission. And this binary fission just involves the division of a single cell, which results in the formation of two cells which are genetically identical. So in the bacteria only reproduction takes place in asexual mode, but in rare case only the reproduction takes place in sexual mode. It is a rare event. There are 5 methods by which the reproduction in the bacteria takes place. The methods are binary fission, first one, then conidia, budding, cyst formation and endospore. Now, what is a binary fission? It is a process that bacteria use to carry out the cell division. It is the process in which the bacteria usually carry out the cell division. And the binary fission, it is similar in concept to the mitosis, which takes place usually in the multicellular organisms. And what happens when the cell divides by mitosis? When the cell divides by mitosis in the body of multicellular organisms, it may result in the organism to grow more larger, and also it is capable of replacing old and as well as burn out cells with the new ones. But in the case of the bacteria, however, cell division is not just a means of making more cells for the body. Instead, it's actually how a bacteria reproduce or add more and more bacteria to the population. So it is but how the bacteria reproduce or how it can add more bacteria to the population. Now next is the steps in the binary fission. In binary fission, a single cell divides into two equal cells. Initially, the bacterial cell, after reaching a critical mass in its structure and cellular constituents, this is the picture of a binary fission, which results in the rapid multiplication of a bacterial cell. So in the human cell, like a human cell, there is a need of the bacterium to copy its DNA. So the copying of the DNA takes place in human cell also. But unlike the human cells, which have multiple linear rod-like chromosomes enclosed in a membrane-bound nucleus, but in the human cells, there is a different character from that of the bacteria. Here, in the human cells, nucleus is enclosed, the chromosomes are enclosed in a membrane-bound nucleus. But in the case of the bacteria, usually they have a single circular chromosome and it always lacks a nucleus. It is not enclosed in a membrane-bound nucleus. However, the bacterial chromosome, it is found in a specialized region of the cell. Which is that region? It is called the nucleoli. And the copying of the DNA by the replication enzymes. That is, the first need is to copy the DNA. And this copying of the DNA takes place with the help of the replication enzymes, and it begins at a spot on the chromosome. And that spot within the chromosome is called origin of replication. The origin is the first part of the DNA to be copied. As the replication continues, two origins move towards the opposite end of the cells. So what happens when the replication starts or continues? The two origins which move towards the opposite end of the cell. pulling the rest of the chromosomes along with them. So along with this origin, the chromosomes along with the DNA origin which move towards the opposite end. And the cell also gets longer, adding to the separation of the newly formed chromosomes. So cell also gets longer by separating towards the both opposite ends, and this may result in the separation of what? A newly forming chromosomes. The replication continues until the entire chromosome is copied. And if the entire chromosome is copied and the replication enzyme meet at the far side. So after the completion of the copying within the chromosome, now the replication stops. Once the new chromosomes have moved to the opposite ends and cleared the center of the cell. So as a result of the replication, what happens? The origin along with the newly formed chromosomes have completely moved towards the opposite end and have cleared the center of the cell. So what happens? There will be a division of the cytoplasm takes place within the center part. So division of the cytoplasm takes place after completely moving the origin as well as the chromosome towards the opposite end. In this process, the membrane it pinches inward and a septum is formed, or a newly dividing wall is formed down the middle of the cell. So septum formation takes place by pinching the membrane inward. Finally, the septum itself splits down the middle and the two cells are released. So septum itself gets splits down the middle. It gets moving towards down the middle. It gets separated and the two cells are released, and they continue to live as an individual bacteria. This is the process. This is the parent cell, mesosome, cell wall, cell membrane. This is the after this origin of the replication, and it moved towards the opposite end. After the so the origin along with the complete chromosomes move towards the opposite end. Septum formation takes place, inward pulling up to the middle, and finally separation of the two cells along with the chromosomes. So these are the daughter cells which are formed as a result of binary fission. The binary fission, it is a rapid process, and the cell undergoes division at an interval of 20 to 30 minutes. So it takes place at an interval of 20 to 30 minutes. The division becomes gradually slow after a certain time. Why? Due to the accumulation of toxic substances and excretion of nutrients. So this binary fission process is taking place within 20 to 30 minutes. And after this division, after some time, the division gets slower due to the accumulation of toxic waste products. Next method of reproduction is taking place in bacteria are conidia. That means conidia formation. And this conidia formation, it takes place in filamentous bacteria like Streptomyces. Streptomyces, it is a type of filamentous bacteria by the formation of a transverse septum at the apex of the filament. So it is forming a transverse septum within the apex of the filament. The part of this filament which bears conidia, it is called conidiophore. After the detachment of the conidia or that filament or conidiophore from the mother cell and getting contact with the suitable substratum, the conidium gets germinate and give rise to new mycelium. So it shows a certain mode of reproduction similar to the fungi. So it's there is a confusion that to classify in which group, either it is a fungi or a bacterium, but finally it was reverted to bacteria because of its filament formation, filamentous nature. It shows certain similarities with the fungi, because in the fungi usually this conidia like this time conidia like things are present in fungi. That's why it gets more similarity with the fungi. So this is the picture of the Streptomyces. This portion. This is called the conidia, and this is called what? Conidiophore. So this is the conidia. This part gets detached from the mother cell, and when it reaches the new substratum, again it results in the formation of a new type of reproduction takes place with the formation of this conidia, and this stalk together is called conidiophore. So this is the type of reproduction which are taking place in the Streptomyces, conidia formation. Next picture is about, next mode of reproduction is called budding. In the case of the budding, the bacterial cell develops small swelling at one side which gradually increases in size. So the bacterial cell which develops a small swelling at one side, and this swelling gets increasing in size. Simultaneously, the nucleus undergoes division. Nucleus means the chromosome part portion undergoes division, where one remains within the mother and one with the cytoplasm, which goes to the swelling. This outgrowth is called, that swelling or outgrowth is known as what? The bud. And this bud gets separated from the mother by partition wall, by forming a wall. Example for this type of budding occurs in Hyphomicrobium vulgare, Rhodomicrobium vanilla. This is the picture of the budding. So this is the swelling portion. This is the budding bacteria, and swelling portion detached, and it forms a new type of cell. This budding takes place in Rhodomicrobium by forming a small swelling at one side which increases in size, and also another remains within the mother cell, and in this the chromosomes undergoes division. One part remains with mother cell, another part move towards the cytoplasm. That's swelling portion. Next is about cyst formation. Cysts are formed by the deposition of additional layer around the mother wall. So deposition of this additional layer around this mother wall, it is called the formation of cyst. These are the resting structures, and during favorable condition they again behave as mother. So these are the resting structures. Due to unfavorable conditions they remain like cyst, and when the conditions become favorable they behave as mother cell. Example for this type are coming under the members of Azotobacter. Next is about endospores. The spores are formed during unfavorable environmental conditions. I think you are familiar with endospores, like desiccation of starvation. As the spores are formed within the cell, they are called endospores. If it is inside the cell, called endospores. Only one spore it is present inside a bacterial cell, and on germination it gives rise to a new bacterial cell, that you already know. So I am not discussing about endospores. It is a type of bacterial reproduction. Then bacterial recombination. In the case of endospores, when the favorable condition comes, it undergoes germination. It has different steps: activation, initiation, activation, etc. The next is about bacterial recombination. The binary fission is an effective way for the bacterium to reproduce, but it is not occurring without any problem, since the cell produced through this type of reproduction are identical. They are all susceptible to the same types of threats, such as environmental changes and antibiotics. So they undergo some kind of changes. As a result of the environmental changes, it affects the bacteria. As a result of this antibiotic accumulation, this affects the bacteria, and these hazards could destroy the entire colony of the bacteria. And in order to avoid this type of conditions, the bacteria can become more genetically varied through recombination. And the recombination involves the transfer of the genes between the cells. And the bacterial recombination is accomplished through conjugation, transformation,